Good morning, everyone, and thank you for being here today. I'm excited to introduce our presentation on the validation of growth promotion study. My name is Dr. S.K. Singh, and I hold a PhD in this field. Over the next few minutes, we'll explore the key aspects of this important study, including its objectives, methodology, and findings. I look forward to sharing this information with you and engaging in a fruitful discussion afterwards. Let's get started. Let's talk about the test principle. Basically, we challenge the media with a small amount of predefined organisms, specifically less than or equal to 100 CFU. The goal here is to see if the media can support the growth of these microorganisms. We expect growth to be visibly detectable within the expected incubation time, confirming the media's ability to promote microbial growth. Additionally, a negative control, which contains no inoculum, must show no growth. This helps us ensure that any growth we see is truly due to the organisms we added, not contamination. Now, moving on to growth promotion testing, or GPT. This is a microbiological quality control test used to confirm that our culture media can support the growth of the microorganisms we're interested in. It's a critical step because it underpins the reliability of our sterility tests, microbial limits, and environmental monitoring. If the media can't support growth, our entire testing process could be compromised. So GPT acts as a validation step to give us confidence in our culture media. Let's take a closer look at some of the key microorganisms that are often referenced in this field. These microorganisms are fundamental because they play crucial roles in various biological processes, whether it's in health, disease, or environmental systems. Understanding these key players helps us better grasp how microbial communities function and interact within different ecosystems. By recognizing their characteristics and behaviors, we can develop more effective strategies for managing their impacts, whether we're aiming to promote beneficial effects or control harmful ones. So, as we move forward, keep in mind how these microorganisms are interconnected with the broader themes of this presentation. Let's walk through an example of inoculum calibration. The goal here is to prepare about 50 CFU, or colony-forming units, for our experiments. First, we start with a pure overnight culture, which gives us a concentrated source of bacteria. From there, we perform serial dilutions to reach our target CFU level. It's important to confirm the actual number of bacteria by back titration, which involves plating a small sample and counting colonies to ensure accuracy. Once we've determined the correct dilution, we adjust it to obtain approximately 50 CFU per plate. Moving on to the test procedure, we prepare our inoculum with a target range of about 10 to 100 CFU, depending on the specific requirements. We then inoculate our media replicates, making sure each one receives the right amount. After inoculation, the plates are incubated under specific conditions. This includes controlled temperature and incubation time, so that the bacteria can grow properly. Finally, we record the recovery, which is the number of colonies that grow, to assess the effectiveness of our process and ensure consistency. This calibration step is crucial because it standardizes our inoculum, leading to reliable, reproducible results across all experiments. Let's walk through how to calculate the recovery percentage. The formula is quite straightforward. Recovery percentage equals the number of CFUs on the test medium divided by the CFUs on the reference or control, then multiplied by 100. For example, if our control plate has 80 CFUs and our test plate has 70 CFUs, then the recovery percentage would be 70 divided by 80 times 100, which equals 87.5%. This percentage helps us understand how well our test medium performs compared to the control. It's important to remember that for non-selective media, an acceptance criterion is generally considered to be at least 50% recovery. This measurement is key for validating our testing process and ensuring accuracy in microbial analysis. Let's talk about the controls involved in this process. First, we have the positive control, which is simply growth on the reference medium. This confirms that our test system is capable of supporting microbial growth under expected conditions. Next, there's the negative control, which involves no inoculum on the test medium. This step is crucial because it helps us check for any sterility artifacts or contamination that could skew our results. To ensure that our inoculum is consistent and accurate, we perform parallel plate counts to verify the CFU, or colony-forming units, of the inoculum. This verification helps us confirm that we're working with the correct microbial load. Now, moving on to acceptance criteria. Typically, we look for visible growth in the test media, which indicates that the microorganisms are present and capable of proliferating. We also expect recovery of at least 50% compared to the control, meaning our test should recover a significant portion of the organisms. Importantly, there should be no growth in the negative control, confirming that our sterility is maintained and there are no artifacts. Lastly, the organism should display its typical morphology, which helps us verify that the growth is indeed from the expected microorganism. All of these controls and criteria are essential because they ensure the reliability and accuracy of our test results, aligning with the overall goal of validating our microbial control processes. Let's take a closer look at this example results table. Here, you can see the key data points we've gathered from our analysis. The table summarizes the performance metrics across different scenarios, making it easier to compare results side by side. Notice how some values stand out. They indicate areas where our approach is particularly effective, while others highlight opportunities for improvement. This kind of detailed breakdown helps us understand the impact of various factors on our overall goals. Connecting this to our broader theme, these results demonstrate the practical implications of our methodology and show us where to focus our efforts moving forward. It's a clear visualization that supports the insights we've discussed so far. Now, let's talk about some common failures you might encounter. One of the main issues is scanty growth, which can really hinder your results. This often happens due to improper media preparation or using media that is old or degraded. If the media isn't prepared correctly or has gone past its prime, the growth you're expecting simply won't occur as it should. The good news is that these problems are usually fixable. The best approach is to re-prepare your media carefully, making sure all steps are followed properly, and then rerun the GPT process. This way, you give your culture or experiment the best chance to grow and develop as intended. So, always double-check your media prep procedures and ensure your media is fresh. Addressing these issues promptly can save a lot of time and prevent more serious setbacks later on. Let's talk about the best times to perform GPT testing. First, it's important to do this with each new media batch. This ensures that every batch meets our quality standards before moving forward. Next, we should perform GPT after the media has been prepared. This helps us verify that the preparation process hasn't introduced any issues. Additionally, it's crucial to test after any storage events or transport. Changes in storage conditions or handling can affect the media, so testing at this stage helps catch potential problems early. Lastly, whenever there's a change of supplier or a new formulation, GPT should be performed. Different sources or formulations can impact media consistency, so verifying quality here is key. Overall, performing GPT at these critical points helps maintain the integrity and reliability of our media throughout the process. Let's talk about documentation. It's crucial to record all the details related to each step of our process. This includes noting the media lot number and expiry date to ensure traceability and quality. We also need to document the organism's strain and source, so we know exactly what we're working with. In addition, recording the inoculum preparation and verification steps helps confirm that our starting materials are accurate and consistent. Don't forget to log the incubation conditions, since factors like temperature and time are vital for reproducibility. Finally, all results should be clearly documented, along with signatures from the responsible personnel to authenticate the records. Proper documentation ties everything together, ensuring our process is transparent, compliant, and ready for review. For further support, please don't hesitate to reach out. We're here to help you implement what you've learned today and to answer any questions you might have. Remember, your success doesn't end here. Ongoing support is just a contact away. Thank you so much for your time and attention. It's been a pleasure sharing this information with you, and I look forward to seeing how you apply these concepts moving forward.